Hello and welcome back to Synapse. Today we'll talk about apoptosis. What is it? It is a pathway of cell death in which the cells activate enzymes that degrade the cell's own nuclear DNA and nuclear and cytoplasmic proteins. So there is some stress on cell's own nuclear DNA. That is, the cell is dictating its own death. So how is this different from necrosis? Is that this is programmed cell death. And another way that it is very much different from necrosis is that in necrosis, there is characteristic cell membrane damage, right? There is damage, so there is leakage of substances into the exterior, which leads to inflammation. But no such thing happens in apoptosis. There is no leakage of substances and no inflammation. And whatever debris are produced because of apoptosis are very quickly removed by the phagocytes before they cause any kind of inflammation. Now, when I say it is programmed cell death, what is programming this? They're genes. Genes are programming this. So let's talk about the pro-apoptotic and anti-apoptotic genes. So pro-apoptotic genes are the ones which increases apoptosis. So here we have P53 gene, BCLXS gene, BAC gene, and BAX gene. These are important examples that we should know. In anti-apoptotic genes, the important examples are BCL2 and BCLXL. And there are a few other genes such as PUMA, BID, and BIM. These can be called the sensor genes because they maintain a balance between the pro-apoptotic and the anti-apoptotic genes depending on the situation. Apoptosis could be physiological or pathological. Let's see a few important examples under both. In physiological, the first important one is about embryogenesis. The apoptosis that happens in embryogenesis holds very important role in order to bring about the final structure of human body. An important example here and a simple example here is that of development of hand. In the beginning, the arm looks more like just a blob. As there is development, there should be removal of this amount of cells so that the digits are formed. So this brings about a structure like this. If this fails to occur, there will be syndactyly. Another important example is about the hormone-dependent tissues. Especially we have the breast and the uterus. This undergo the endometrium of the uterus. This undergoes cyclical changes with the menstrual cycles and also post-pregnancy. As the hormones that maintain either the endometrium or the breast tissue, as the level of those hormones fall down, these tissue also regresses. Then it's about the TLC, that is total leukocyte count. Let's say a person had infection. So his TLC would have gone high. It would have increased in number. After the infection comes down, after the infection is all gone, the TLC has to come back to the normal value. And here is where the apoptosis plays an important role. Now let's look at the pathological apoptosis. Whenever there is DNA damage, let's say because of cytotoxic anti-cancer drugs or because of radiation, extremes of temperature, any factor which causes DNA damage is going to put the cell under risk. There could be mutation. So initially the cell will try and repair the DNA damage. But if the damage is too much and it cannot be repaired, then it has to undergo apoptosis. Because we cannot afford to give rise to some kind of mutations. It may lead to malignancies, etc. Next is about viral infections. Viral infections are intracellular infection, right? We know that. So the only thing and the effective thing that we can do against viruses is to kill the cell that it has infected. So here is where apoptosis plays a role. A point of special mention here is about viral hepatitis. In viral hepatitis, we see this characteristic structure called as Councilman bodies. Councilman body. This is a point of high yield. Next example here is retinitis pigmentosa. Here there is apoptosis of rods, and we know that this pigment is important for vision in dim lights, and thus apoptosis of this will lead to night blindness. Let's look at the pathways by which apoptosis occurs. We have intrinsic pathway, extrinsic pathway. At the end, what it is doing is that both the pathways are coming and activating or increasing the activity of caspases. Why is it called caspases? It is cysteine protease. It's a cysteine group containing protease. It acts at the site wherever there is aspartic acid. Right, in the proteins, if this is aspartic acid, this is where they're going to cleave, and ase is going to again signify that it is a protease. That is what caspases mean. They're a group of proteins, so proteases, that are involved in apoptosis. So now let's look at how intrinsic and extrinsic pathway come and activate these caspases. Let's look at the intrinsic or the mitochondrial pathway. Here we have our mitochondria. In mitochondria, we have a few substances that have apoptotic properties. An important example of such substances is cytochrome C. But this normally cannot leave the mitochondria because the channels via which they can are kept closed or they're blocked by anti-apoptotic proteins such as BCL2. Now let's take a situation where there is decreased growth hormone or decreased stimulus for growth. When there is any such situation where apoptosis should occur, there is decreased BCL2 and increased BAC and BACs. We already know that BAC and BACs are pro-apoptotic genes. Now, because there is decreased expression of anti-apoptotic genes and increased expression of pro-apoptotic genes, this finally leads to leakage of cytochrome C into the cytoplasm. This cytochrome C, which enters the cytoplasm, is now going to activate APAF1, apoptosis activating factor 1. This further activates caspase 9 and 10. These caspases are called the initiator caspases for intrinsic pathway. They're called initiator caspases because they further go and activate caspase number 3, 6 and 7. And these caspases are called the executioner caspases. What is it executing? Or how is it executing the apoptosis job? It is activating two sets of enzymes. One is nucleases, which bring about the degradation of the nuclear material. And second is the proteases, which bring about the degradation of the protein material. Now that the nuclear and the protein material of the cell is degraded, the cell can definitely no longer survive. So this brings about apoptosis. Next pathway is extrinsic pathway. So here we work with few receptors. On our cell membrane, we have a few receptors, and the example for these receptors could be Fas molecule or TNF receptor, tumor necrosis factor receptors. These are some of the important examples for such receptors. These receptors have an intracellular domain, and this is called as FADD. that is fast associated death domain. This called as the death domain as they mediate the cell death, right? So now let's say we have the antigen, that is, if this receptor I'm talking about is fast molecule, then this would be fast ligand, right? Or if I'm talking about TNF receptors, then this would be TNF alpha. Now, when the respective protein binds to the receptor, there will be trimerization of the receptors. So here I'm showing trimerization of the receptors. What is the importance of the trimerization? This activates the FADD component. The activated FADD converts pro-caspase 8 to caspase 8. Basically, it is activating caspase 8. So what does that mean? It means that caspase 8 is the initiator caspase in case of extrinsic pathway, in contrast to caspase 9 being the initiator caspase in the case of intrinsic pathway. Now, caspase 8 does exactly what caspase 9 does in intrinsic pathway. That is, it activates caspase 3, 6, and 7, which are the executioner caspases. This further activates nucleases and proteases, which bring about degradation of the nuclear material and protein material respectively and bring about apoptosis. A high yield point that you would want to know here is that fast ligand is also called as CD95. Another important point is a hallmark feature of apoptosis. Hallmark feature here is chromatin condensation. In contrast to necrosis, in necrosis, the hallmark feature is cell membrane damage. But in apoptosis, you do not see cell membrane damage. And here, the hallmark feature is chromatin condensation. And with respect to nuclear changes, in necrosis, you see karyorrhexis, karyolysis, and pycnosis, and not chromatin condensation. Next thing we're interested to know is how do we detect apoptosis? So here, first method is we have something called tunnel technique. What does tunnel stand for? Terminal UTP nick and labeling technique. So now this technique, the name itself explains a lot, right? Terminal UTP nick and labeling. So here the stain that we use is UTP, which is brown in color. And this binds to the 3' end of the DNA material. Now let's take a scenario where we have DNA material which is not broken. So we have the 5' end here and 3' end here. And another scenario where this DNA material is broken down into several fragments. And each fragment will have its own polarity. 5', 3', 5, 3, and so on. Now, if I add this stain, that is UTP, in the two scenario, where do we see darker color? Definitely, in the second scenario, you're going to see darker color. So when you see that there is a darker staining, it means that there is DNA breakdown that has occurred, that is DNA fragmentation. So it's one way you can identify apoptosis. Another way you can do this is by gel electrophoresis. So in this, let's look at how it is for normal DNA material in case of necrosis and in case of apoptosis. In case of a normal cell or normal DNA material, you're going to see only one band as the DNA is not broken down. In case of necrosis, you will see smear pattern. This is called smear pattern. This is because there is DNA damage seen in necrosis. In case of apoptosis, we see step ladder pattern. Again, because of DNA fragmentation. And this is very characteristic, and we really must remember this. Now, another way that we can recognize is that a cell which undergoes apoptosis, this is a protein which is normally expressed on the inner aspect of the membrane. Now let's say it undergoes apoptosis. Now it expresses this protein on the outer side. Its actual functionality is that it is going to attract phagocytes. Going to attract phagocytes, which will come and eliminate the cell as quickly as possible so that there is no resultant inflammation. But now we can use a dye which uses this protein as a target, and we can identify apoptosis. So such dye that we are talking about is annexin 5. Another method you could identify apoptosis is look for cytochrome C levels in cytoplasm. So it's definitely high in case of apoptosis. And you could also detect the condensed chromatin. As already mentioned, condensed chromatin is a hallmark feature of apoptosis, and thus it can be helped in identifying apoptosis. Before we end our discussion about apoptosis, it's necessary that we talk about necroptosis and pyroptosis. So about necroptosis, it's a mixture of necrosis and apoptosis. How? It starts like how apoptosis starts. Here we can see the receptors, and it is here are the receptors, and here we have their ligands binding, which leads to trimerization. And this trimerization in this condition, it does not lead to caspase activation, but instead it causes free radical formation. This free radical will further go and cause damage to DNA material, proteins, and lipid. And once you damage all these important components of the cell can no longer live. So this is necroptosis. What's important about necroptosis is that it is, as understood, it is caspase independent programmed cell death. While apoptosis is caspase dependent programmed cell death. What are the examples where we see necroptosis? First, we see it in Parkinsonism. We see it in pancreatitis. And very important is immunity against cytomegalovirus. In apoptosis, I already mentioned that viral infections are basically combated by apoptosis. But why for cytomegalovirus we need necroptosis? It's because cytomegalovirus is a smart virus. It's going to inhibit caspases. And thus, apoptosis, which is caspase dependent, will not be effective against cytomegalovirus infection. Thus, necroptosis comes into picture here. Last thing now is pyroptosis. We know by the word that it is associated with fever, and there is some component of apoptosis. So let's say in an example where there is bacterial infection, This leads to inflammasome formation, intracellular inflammasome formation, which leads to activation of caspase 1 and 11, which further leads to interleukin 1 formation. And we know that interleukin 1 is associated with fever. And thus, intracellular inflammasome formation, where there is caspase 1 involved, leading to fever, is called as pyroptosis. Here we end our discussion on apoptosis. If you found it useful, please do like, share, and subscribe.